Welcome back to the chemical foundations of life. We were in the previous class, we were dealing with the chemical foundations of life. Now we are continuing. You see, the complexity of the biological system, the biochemical system, is increased by the versatility of carbon bonding. We have studied in your lower classes that the valency of carbon is 4, and between two carbon atoms there can be single bonds, there can be double bonds, or there can even be triple bonds. But in biochemistry, carbon carbon triple bonds are very rare in biomolecules. You see the different types of bonding by sharing. I see all these bonds are actually covalent bonds. By sharing one electron each, a carbon hydrogen single bond is produced. I hope you know what is meant by covalent bond and ionic bond. These bonds are covalent bonds formed by sharing of electrons. By sharing one electron each, a carbon oxygen single bond is produced, or by sharing two electrons each, a carbon oxygen double bond is produced. You might have seen these bonds in aldehydes and ketones. Remember, carbohydrates are either aldehydes or ketones. Again, carbon nitrogen single bond, as you see in the case of amino acids, carbon nitrogen double bond is there, carbon carbon single bond is there present in most of the biomolecules, carbon carbon double bond is present in some of the fatty acids, etc. So this is a common feature in biochemistry. And another feature is that the geometry, they can have a characteristic tetrahedral arrangement of the four single bonds, and they have the freedom of rotation. You see, along this single bond, this carbon can rotate. And double bonds are actually shorter, and rotation is not possible around a double bond. For example, this configuration is fixed for these atoms because this double bond does not permit any rotation of this carbon on this axis, and the double bonds are shorter also. These are the functional groups which we see commonly in the biomolecules. You see, CH3 group is methyl group, NH2 is amino group, and ethyl group is there, and amide linkage is there, as you see in the case of some amino acids, etc. Phenyl groups are there, and a guanidine group, carbonyl, imidazole, and a carbonyl group. This is ketone. Sulfhydryl groups are there, carboxyl, disulfide. All these functional groups are responsible for the maintenance of the biochemical system. And here in all the cases you can see R group. In all the cases you can see R group. What does it mean R? R simply means any substituent. Sometimes you can see here there is an R1, here there is an R2. What's the difference between R1 and R2? They are two different substituents. R1 and R2 are two different substituents, and these are the functional groups which we commonly see in the biomolecules. And these are some other substituents. Hydroxyl group is there. This is commonly there in the case of carbohydrates, glucose, fructose, and all the types of carbohydrates. You can see this alcoholic group.